Right, ladies and gentlemen, in this presentation you're in for a treat, because it's a short one. So here's what's going to happen. We're going to talk a little bit about what goes into the process of actually reading the genetic code. All right, so it's short, it's sweet, but it's actually incredibly important. I need you to understand that you have to sort of think about DNA as if it's a language. But it's an interesting language because it's a language that's written in only 4 letters. And it's not a language that has words. It's not a language that allows us necessarily to communicate with others. Instead, it's a language that contains all the information about proteins, about polypeptide chains. It contains the information about their structure, and structure equals function. Now, the 4 letters that DNA is written in is known as nucleotides. All right, so you've got a language that's written with only 4 letters called nucleotides, and at this point, you should know what they are: adenine, thymine, cytosine, guanine, A, T, C, and G. Okay. Now, the thing about proteins is that we've got this language of DNA that gives us the information about proteins. But the DNA language is written in 4 letters that we call nucleotides. Whereas the protein language, all the pieces that give proteins their structure and therefore their function, there's actually 20 letters because all of your polypeptide chains are built of 20 different amino acids. So the big question when it comes to using DNA to generate protein structure is, how do you read a 4-letter language and have it become a 20-letter language? That's the real question here. So, scientists pondered this for a while. There's actually some information in your book about the whole research aspect of it. But basically, they went through a series of suppositions. They basically said, well, what if every one of the nucleotides codes for one amino acid? Well, there's 4 nucleotides, so that means that at most you could code for 4 amino acids. That's not enough. There's 20 amino acids, so it can't be a 1-to-1 relationship. So then they said to themselves, all right, so what if it's 2 nucleotides gives us 1 amino acid? So what it's saying is, if you're looking at a DNA strand, you look at the nucleotides in groups of 2. So AC, then GT, then AA, then TT. And if each of those groups of 2 gives us 1 amino acid, then that's a total of 16 amino acids. Now, you may ask yourself, how is it that it's 16 amino acids? And it's important for you to realize that the reason it's 16 is because there's 4 nucleotides. And since we're looking at all the possible combinations of 2, it's 4 to the 2nd power. So 4 times 4 is 16. Okay? So 16 isn't enough. It's close, but it's not 20. So then, obviously, the next logical step is to say, well, what if we read the nucleotides in groups of 3? So ACC, CGC, ATA, TTA. So that every 3 nucleotides we see on a DNA strand gives us the information for 1 amino acid. Well, that sort of overshoots the mark a bit, all right? Because take a look. There are 4 nucleotides, and we're looking at them in groups of 3. So all the possible variations is 4 to the 3rd power. Well, 4 times 4 is 16. 16 times 4 is 40. 24 gives us 64. Well, that's certainly enough, but it's also more than enough. But there really isn't any options. It's not like you can cut the nucleotide in half. So that's when scientists realized that what's actually going on here is to translate the information contained in DNA to the structure and therefore the function of proteins. You have to look at them in groups of 3. Groups of 3 that we call codons. So codons are 3 nucleotides that provide the information necessary so that your cellular machinery, which we will learn later is things like transfer RNA, messenger RNA, ribosomes, different polypeptide factors that aid in that process, to create amino acid chains in the right order. And if amino acid chains are in the right order, then that means they have the right structure. If they have the right structure, they have the right function. And if you change the order of the amino acid chains, you change the function of the amino acid chains. And so that's how DNA provides the information for polypeptide structure. by creating the order the amino acids are joined in. Different order of amino acids is different structure, which is different function. And DNA in the codons contains the information for the order the amino acids are joined in. This table right here, which you need to become very familiar with, is the genetic code. Basically what this is this is the codons, and you'll notice on this codon table, we don't have T, we have U. And think about this for a second, all right? If I have a DNA strand, the DNA strand might do this, A, T, G. Then I make a complement, so this right here is DNA. Okay, so if the DNA is ATG, and then I make a complementary strand of messenger RNA. All right, so here's my messenger RNA. Well, if it's complementary, so this messenger RNA reads the DNA and it leaves me with U, because remember, there's no T in RNA, it's uracil instead, A, C. All right, so immediately right here you can see why there is a U on this table and not a T, because this table is talking about the codon and anticodon relationship. But see, that's an interesting thing about this, is we're not done. All right, after the messenger RNA is formed, the sequence of amino acids is determined by anticodons on tRNA molecules that actually complementary bind to this mRNA. So what that means is that the tRNA is another complementary matchup. So U becomes A becomes U, and C becomes G. Here's the most important thing about this. The reason that this table is actually the genetic code, take a look at this. The anticodon on the tRNA is the same as the DNA sequence. The only difference is that RNA does not have thymine. It has uracil instead. So this mRNA, this transcription process, actually allows us to make sure that the anticodon is identical to the original DNA strand. So let's talk about how you read this. Wow, this table is incorrect. Okay, so you read this table by starting with this column right here, okay? So you read this table by starting with this column. So if you're doing AUG, you would go A, and you would know that your answer is somewhere in this row right here, okay? Because you start on the left. So over here is where you go first on this column. And then second, okay, you go right here. So AU. So we know that we're in this row. I'm sorry. Your answer from the first column is in this row right here. And we also know now that it's in this column. All right. Well, where those two things intersect, so literally right here, we know that our answer is one of these four. Now, let's be honest. You could go over here for the third base and be like, oh, it's AUG, so it's this one. But the majority of people simply just go, oh, it's in this column and this row. Let me look at these four and figure out which one it is. Okay, so, you know, in this case, we know that the answer is actually this one right here, AUG. So, first column starts on the left. That gives you your row. Second is on the top. That gives you your column. And then you just go to the box where they intersect and find which of the four your answer is. All right, that's it for how you read the genetic code. Told you this one would be short.